I'm Chris McAleer, principal scientist here at Hesperos. Can you hear me okay? I apologize in advance. I'm part businessman, part scientist, so there will be shameless plugs here, so forgive me for that. I'll be talking today on the classical complement pathway inhibition in the human on a chip model of autoimmune demyelinating neuropathies. And Mike alluded to this earlier today, and I think it's really apropos for this case studies of MPS, because to our knowledge, it is the first microphysiological system data to be included in IND that's been allowed to go to phase 2 clinical trials. So we started with a company called True North Therapeutics, a small biotech company, like happens in all the biotech world. They got gobbled up by Bioverativ halfway through this process. And then shortly after the initial phase of this finished up, Bioverativ got gobbled up by Sanofi. And then it sat on a shelf for a little bit, and they finally decided to put this into an IND. Before I get started, though, I do want to make sure I give credit where credit is due. I want to thank John Wayne Rumsey, Dr. John Wayne Rumsey. He was a scientist leading a lot of the oversight of the day-to-day operations. He's now the director at the Duke Vaccine Institute, and Case Lawrence, a research associate who did most of the grunt work for this. So one of the really nice things I like about these talks is that I don't have to convince you guys about the microenvironment, surface chemistry. I don't have to convince anybody here about the design of MPS, right, or the cell types that are there. But I do want to talk shortly about, like the MPS design is important, the microenvironment and the surface chemistry is important. We use a lot of non-biological self-assembled monolayers. It allows us to do photolithographic patterning of the cell surfaces for spatial dimensions of cells for cell-cell interactions, also like the spatial organization of that for cell maturation that Mike alluded to and some of the cardiomyocyte data that we have earlier. And for us, it's really important. We use, as Mike alluded to, also we use a serum-free medium, right? It's chemically defined. It is a little bit expensive, to be truthful, right? But we look at that and say, if we can define the nature of all the ingredients that are there, and we know the interaction of the cells to the surface, to the medium, right, in the microfluidic device that we have, right, both computational fluid dynamics, right, how that affects, shear stresses and flow rates, and how that affects nutrient utilization, right, we have a better understanding of how the system works. And shameless plug for Hesperos, we are a vertically integrated company, so we do in-house have everywhere from a CNC machine and like people who manufacture them to computational fluid dynamic modeling to Python coders to surface chemists, analytical chemists, and a bunch of biologies and engineering team led by Dr. Long. If you see him in the hallways, you can stop and say hi. So I'm sure that many of you who have seen any talks at Hesperos have heard functional readouts over and over again, right? And I'm going to beat it in again, right? We really pride ourselves on the ability to measure these functional parameters, right? And so I think you alluded to earlier today, there's functional parameters many times that, for our devices and depending on how you design them, precede cell death by quite a bit of time. We've even seen the functional parameters, say conduction velocity of the heart, actually precede troponin or creatine kinase release into the biomarker release into the medium. Right. And so they're really sensitive measures of cellular function. And for us, it's really important because, we also pride ourselves on this ability to measure not only acute dosing, but repeat dosing, right? Or progression of disease over time, say in Alzheimer's or ALS, right? As you press in, some of this data will be in some posters that you'll see later, right? Where you can measure the differences in the genetic makeup of the cells, right, and how it progresses over time and the onset of disease. And we think that it will augment the biomarkers that are needed, right? We don't have to predict what the biomarkers are going to tell us about function. We can measure that function directly. And so we were approached by True North Therapeutics to study multifocal motor neuropathy and chronic inflammatory demyelinating polyneuropathy. And they said, do you think you could build us a model of this? I said, sure, let me go Google what MMN and CIDP are, because I don't know. So for any of you out there who don't know what MMN and CIDP are, they are very similar disease phenotypes that for our purposes we're looking at only the motor neuron portion of it. They do exhibit different clinical manifestations, whether the CIDP is symmetric and affects both motor and sensory portions, where multifocal motor neuropathy is asymmetric and only affects the motor unit. And so what the common features of these is that you get persistent conduction block of the neurons, and that a lot of the patients are shown to have GM1, IgM, and IgG antibodies circulating. And the theory behind this is that the autoimmune antibodies, the GM1 mediated IgG, IgM antibodies, will bind to neurons. They believe at the node of Ranvier, and that binding of the IgG, IgM will induce the complement cascade and then affect the motor neuron conduction. And so True North Therapeutics had a compound that was a C1s inhibitor, and they wanted to look, it's called TNT-005. I'm assuming it was the 5th generation of that. And they wanted to see whether or not that if it would block the complement deposition, and B, if we could measure conduction block and its ability, the patient serum sample's ability to modulate conduction velocity of the neurons and their molecule's ability to ameliorate that conduction block. And so we thank you, Jeff Guptill from Duke University, who gave us multimodal photoneural neuropathy and CIDP patient serum samples. So the first thing we wanted to do was to ensure that the patient serum samples that we got, there's a lot of the patients go IVIG therapy that's supposed to remove a lot of these autoantibodies. We wanted to ensure that there were GM1, IgM, and IgG antibodies in each of the patient serum samples that we collected. We got an ELISA to do that, and this is just quantification of the differences in the IgG, IgM antibodies in each of the patient serum samples that we received. And so before we move further, we wanted to test the utility of the system. Can we ever even measure complement deposition? So in a very controlled environment, before we wasted a bunch of very rare patient serum samples, we wanted to test a secondary only antibody for complement deposition. We did a normal human serum control with 0.05% lyophilized complement, and a positive control was an anti-GM1 antibody where we added complement. And we looked at both Schwann cells and motor neurons, and we can see anti-GM1 mediated complement deposition both in Schwann cells and motor neurons. And if you can tell, there's a lot more in the Schwann cell portion. And so with that confidence that the system will be able to measure GM1 mediated complement deposition, we took the patient serum samples, and we wanted to ensure that the patient serum samples would deposit IgG and IgM antibodies onto both Schwann cells and motor neurons. And they do, more so on the Schwann cells. And you get a lot more of the anti-IgG, which is clinically what you see. And so the next question was, do these IgG and IgM GM1 mediated antibodies activate the complement cascade from the patient serum samples? And so again, we took monocultures of Schwann cells and motor neurons, and indeed, both in Schwann cells and motor neurons, you get C3b and C5b through 9. And in the Schwann cells, you get quite a significant deposit of the C5b through 9 complement deposition. And so before we used a lot of the TNT-005 antibody and more patient serum samples, we wanted to ensure that through our control mediated GM1 system, that we can get TNT-005 blockage of the complement deposition. And when Dr. Rumsey first showed me this data, I said, clearly you have mislabeled your images, because there's no way that you got this much blockage of the complement. So I do want to state that this is blockage of complement deposition and blockage of conduction block. They're co-administered, both the patient serum sample and the TNT-005 antibody were co-administered simultaneously. At some point, we'd like to look at the ability to reverse this phenotype, but we thought the most staggering results at the very beginning of this project would be co-administration and blockage. And so I said, repeat it. They repeated it, got the same answer. I said, okay, well, I feel comfortable then to move forward with the patient serum sample. And these are two representative images of the complement deposition. And these are co-cultures of both Schwann cells and motor neurons together, that you get quite a bit of complement deposition in all the serum samples. And TNT-005 was able to reverse or ameliorate that complement deposition. anywhere from 88% all the way up to 97 or 98% reduction in complement deposition. And so Bioverativ, well, at that point it was True North Therapeutics, was extremely happy about this data. And they said, okay, great. So you can block complement deposition, but what does that have to do with conduction block? Do we actually see any conduction block of these motor neurons with this complement deposition? Okay, so we built a microphysiological system to study this. And so an SU8 mold and a PDMS design, it was very simplistic. To guide axons, we'll put motor neurons on one side, Schwann cells on the other, allow the Schwann cells to migrate into the tunnel, allow the axons to go through the tunnel, and you'll get Schwann cell motor neuron communication inside that tunnel. I'm sure that some of you guys are going to ask us later. We tried to measure the extent of myelination in the tunnels, but trying to take the tunnels off or trying to slice them or trying to vacuum antibodies through caused a lot of damage to the cells. And so at this particular point, we're still trying to figure out how you look at the extent of myelination within the housing design. But right now, all we know is that there is either trophic or physical interaction of Schwann cells and the axons within these tunnels. If you want to look at any of the myelination work that we have done, Akash is giving a talk later today, or a poster later today, poster number 27. You can look at our myelination model, both using primary human Schwann cells and iPS-derived Schwann cells. And so, as you can see, is there a pointer? This thing? As you can see here, we aligned the PDMS tunnel over the electrodes, and you can see multiple axons that run through. Because of those multiple axons, you do get so what we wanted to really look at was at least 3 microelectrodes in a row where you get a signal that propagates across, right? And since we know the spatial distance between each of those electrodes, we can measure the conduction velocity of the neurons. And because we have a range of axons that are there, how do I go back? We have a range of the conduction velocity of those axons. And what we found is that the average conduction velocity is about 0.2 meters per second, which is similar to in vivo unmyelinated small diameter axons. So we were pretty comfortable that they're physiologically relevant. And so we wanted to look at both the negative control and the positive control, right? Just normal human serum samples with a TNT-005 isotype or the TNT-005 inhibitor. And what we saw is that it really doesn't matter. You get some fluctuation up and down of the conduction velocity with a negative control. But in the positive control, you get a massive reduction in the neuronal conduction velocity and also in the action potential firing frequency, and that the TNT-005 was able to ameliorate that phenotype. And so then we quantified that both in the negative control group where there was no difference between the isotype treated or the TNT-005 treated, and both in the firing frequency and the conduction velocity. However, in the isotype treated, you get approximately 80% decline in both frequency and conduction velocity with the isotype treated, and that is statistically significantly ameliorated by the presence of the TNT-005 in both the positive control. And you sort of see a graded response with it. We took all 15 of the patient's serum samples and lumped those together to quantify, and you get about a 50% reduction in both frequency and conduction velocity across those patient serum samples, and that also is ameliorated by the TNT-005. And so that data was put together in a package with Sanofi to go into an IND that was submitted in December of 2020 and allowed to go to clinical trials in April of 2021 and is currently ongoing. And so what I hope that I showed here is that we build a system to model a subset of clinical features of MMN and CIDP, and that their proprietary antibody TNT-005 was able to block both the complement activation and deposition as well as conduction block. And I think we're working on this model at this particular point has therapeutic value in quite a few rare diseases like Friedreich's ataxia or Charcot-Marie Tooth syndrome, right? Or even Pompe disease. And in some parts, we're looking at its application towards ALS. And so I will open up the floor to any questions that you might have. And shameless plug. If you like anything that you saw today from Mike Schuler or Jay's talk yesterday or anything that I said today, we are currently hiring quite a few PhD level scientists, innate immunologist, a liver biologist, and list goes on. We do have a booth that you can go. And if you have any wants or needs, we pride ourselves in our ability to build whatever you want. I didn't know anything about MMM or CIDP when we started this, and here we have an IND. So we can build anything that you ask us to.
I have a question for the speaker.
So the technical question is the images you show for the MAC system, right, of the complement. So you show just one color. So you're using one specific antibody that identifies all the components of the system.
So we actually stain for both. There's an anti-C3b antibody and a C5b through 9. It's a polyclonal for that.
Okay.
What we found was we initially were upset because we couldn't see any C3, but if you, and this data is not in there, if you stop the systems earlier and fix them and stain them, you'll actually see the C3. I think the way that the system is designed, when the C5b through 9 complex gets laid down, it blocks the ability to really see the C3. So a lot of it you'll see was in red because that's the C5b through 9, right? Downstream of the complement cascade, right? But if we stop that early, you see more of the C3. And so we have that data, but it's initially we were concerned, but temporally speaking, it makes sense.